Hello everyone, my name is Vincent Beliveau and together with my colleague Anita Johansson, we will present our work on the creation of a human brain atlas of in vivo pre-synaptic density. The study was performed at the Neurobiology Research Unit of Rigshospitalet in Denmark.
Synapses are the foundation of brain function. This is where vesicles containing neurotransmitters are released into the synaptic cleft, where they will then bind to receptors on the postsynaptic neuron. This is visualized here in the left figure. The right figure shows an actual synapse visualized with electron microscopy, where the vesicles are visible as round shapes. In the membrane of the synaptic vesicles are embedded multiple proteins, one of which is the synaptic vesicle glycoprotein 2A, or in short, SV2A. SV2A is ubiquitously expressed in gray matter regions of the brain and is therefore seen as a marker for synaptic density. Using radioligands such as carbon-11 labeled UCB-J, which is specific to SV2A, it is possible to study synaptic density in vivo with positron emission tomography. The very first study to investigate SV2A in vivo in humans was performed by Rich Carson's group at Yale in 2016. Brain atlases are becoming increasingly useful as reference tools for new studies and for comparing healthy brain function or disease states. A very good example of the usefulness of brain atlases was recently provided by Hansen et al. 2022, where atlases of multiple molecular targets were used to investigate the association with brain function and disease. As we have collected multiple SV2A PET scans as part of ongoing studies at NRU, we decided to leverage this data and create a new high-resolution atlas of SV2A density in the human brain.
To create the new atlas of SV2A density, we used a surface and volume-based processing of the PET data, which was previously developed in collaboration with Doug Grieve from the Christopher group and used to create atlases of the serotonin receptors and the serotonin transporter, as well as the GABA-A receptor. Our cohort is composed of 33 healthy volunteers, 17 females, 16 males, with a median age of 24 years old. So this represents a relatively young population.
Each participant underwent PET scanning where they got an intravenous bolus of the radioligand UCB-J. The radioligand will then bind to SV2A, and when it decays, gamma rays are emitted and picked up by the PET camera. In our study, we scanned for 2 hours with a high-resolution research tomograph PET scanner, which has a point-to-PET function of 1.4 millimeters in the center of the field of view. From the recorded data, 3D dynamic frames are reconstructed and further pre-processed and used for quantification. In addition to the PET image, we also acquired a T1 weighted image from a 3 Tesla MR scanner to provide additional anatomical information.
Here is an overview of the preprocessing approach used for the PET and MR data. Briefly, the reconstructed dynamic PET data is motion corrected and a time-weighted average image is created to compute the alignment between the PET space and the MR space. Christopher is used to process the T1 weighted image and obtain a model of the cortical surface and compute the alignment to MNI 152 space. The dynamic PET data is then resampled onto the standard cortical surface and in the MNI 152 space. The data is then surface and volume smoothed, respectively. Regional time activity curves for the cortical and subcortical regions are extracted from the non-smoothed data. Smoothing is an essential step in the processing of PET data in order to reduce noise before performing kinetic modeling. One of the main advantages of our approach compared to traditional volume-based processing of PET data is that we smooth the cortical data in the surface space rather than in the volume. As exemplified in this figure, surface-based smoothing has a few advantages compared to volume-based smoothing. Firstly, due to the spatial folding of the brain, 2 points located far apart on the cortical surface may be located very close in the volume space and will become averaged together when performing volume smoothing. Secondly, by disregarding the spatial organization of the brain, we end up mixing up signal from different tissue types. Effectively, volume-based smoothing exacerbates paucity volume effects in the PET data and reduces spatial resolution. To quantify SV2A in the brain, we used a one-tissue compartment model with metabolite-corrected arterial input function.
PET does not provide absolute measures of protein density. However, autoradiography allows for quantification of absolute protein density from postmortem brain tissue while retaining high spatial resolution. To perform autoradiography, the brain is cut into very thin slices and then incubated with a solution that contains a high concentration of our radioligand, which will bind to our target. The slices are then washed to remove excess radioligand, and then the signal from the slices is captured with imaging plates, which are scanned to obtain the final image from which absolute protein densities can be obtained. We quantified SV2A in the brains of 7 donors and supplemented our data with previously published autoradiography results from the Karolinska group. We observed a strong association between the autoradiography and our PET data and used a linear relation between the two to convert our PET data to absolute densities. However, it's worthwhile to point out that some regions diverge between the two methods, such as the putamen and the hippocampus. Here we can see the average SV2A density maps on the cortical surface. We observed that the posterior cingulate, precuneus, and temporal cortex are the regions with the highest SV2A density, whereas the sensory motor cortex and the lateral occipital cortex are regions with the lowest SV2A density. Subcortically, we observed the highest SV2A density in the putamen. In that region, the density did not appear to be homogeneous, and higher density was observed in the anterior part of the putamen. The pons cerebellum, hippocampus, and pallidum were the regions with the lowest SV2A density.
To gain novel insight in the expression of SV2A protein and its localization, we compared regional SV2A density to mRNA levels obtained from the Allen-Newman Brain Atlas. We observed no significant association. Pioneer work using carbon-11 UCB-J has been previously performed by Finema et al. 2016. Recently, an average population map of these data have been released within the NeuroMaps Python package. Beyond small differences in demographics and the model used to quantify the PET data, the main difference between the maps from NeuroMaps and ours is the type of preprocessing used, that is, surface or volume-based. As we can see in the upper left figure, at a regional level, there is a strong correlation between our results and those of NeuroMaps, although we can observe some large differences in individual regions. However, when we look at the data projected onto the cortical surface, we can see striping patterns in the data from NeuroMaps, which are typical of increased partial volume effects. Strikingly, we can see a strong correspondence between these patterns and the curvature of the surface model, which seems to exhibit a negative linear association as observed in the upper right figure. In our data, the striping patterns are much less prominent, and there doesn't seem to be an association with the curvature of the surface model.
There are interesting associations to be observed between SV2A distribution and what we know about the organization of brain function. For example, low SV2A density is observed in the primary motor and sensory cortex. When comparing with the human homunculus, there appears to be lower density in the medial areas representing larger limbs compared to the more lateral areas associated with the face and mouth. It is not exactly clear to us how to interpret this pattern, and more rigorous investigation would definitely be needed to understand what this means. Another interesting observation is that the distribution of SV2A in the putamen appears to vary corresponding to projections from different functional areas in the cortex. For example, here we can see that the anterior part of the putamen, which has the highest SV2A density, receives projections from cortical areas associated with higher cognitive function, whereas the more medial and posterior part, with lower density, receives projections from the primary motor cortex.
Our atlas of in vivo SV2A density will soon be available on NRU's website, and we look forward to see how people are going to make use of this new tool. Thank you for your attention.